Right, so building on this, we know that mutations occur naturally over time. Sure, there's environmental factors or other genetic factors that can increase how frequently mutations actually occur, but we know that if we look distinctly along, enough millions of years, we naturally see that organisms over time generate mutations. It is a natural course of humanity and every other species' existence, is that they just will and always will occur. But what happens in this is if mutations occur enough and they're not destructive to the organism, so they're not something that actually completely makes a protein cease function or removes a capacity to do an essential job, what happens is those mutations bring rise to evolution. So they bring rise to organisms looking or appearing slightly differently, being able to create slightly different proteins which serve slightly different functions. And if that happens naturally over time, what we have a look at is when we can take a step back and look at the amino acid that is then within a sequence, which the amino acid is made up of 3 bases of DNA. If an amino acid is changed because there's been a mutation and that change was not detrimental to the structure or function of the organism's existence, essentially, we're going to start to see that more closely related organisms will have less mutations actually between their amino acid sequences because they're going to have less changes in the overall amino acids because there's less changes in the DNA itself. So this is really that basis of how we can use sequencing to determine how closely related we might be to a dog, a bird, a frog, a lamprey, or a monkey, for example. So then we have a look here. We can now start to look at key proteins that if they were changed, what would happen is that would be detrimental to the organism. And so this cytochrome C protein is one that is super, super important, that it doesn't actually have a large amount of, doesn't have too much variation, or the variation that it does have, it still needs to be able to conduct the same job. And cytochrome C is a really important protein for all living organisms, essentially, because it is involved, all living organisms that have a mitochondria, because it's involved in aerobic respiration. So because it's a very commonly found protein that is actually made, what that means is it means that we can have a look at all organisms that contain that cytochrome C protein and then look at the comparison along the amino acid sequence to see how many changes there might be, with the cytochrome still being a functioning protein. So obviously if it was a non-functioning protein, that would probably be disastrous. That organism would not be able to conduct aerobic respiration. But you can see here, if we look at where it says 1, for example, you can see all the way down the line, all of those organisms at the 1 or the first amino acid contain the exact same amino acid. We can see in the second sequence of the amino acid along the protein, you've got a horse and a human contain the same one, and so does a monkey. However, we see variation that there's the GLU, the ALA, the GIN, and the VAL in those other organisms, which infers that we're not as closely related to chickens, to frogs, to sharks, and to rabbits as we are to monkeys and horses. We keep going down that line and we can see in the third amino acid, the PHE there, and we compare that to humans, is different. It's the TYR amino acid, so there was a mutation at some point along the evolutionary line. But we can see that the monkey also has the TYR amino acid. So this now starts to let us be informed that humans and monkeys are more genetically or closely related than humans and horses, for example, which means that we diverged or became distinct species much, much later in the piece, or, not as long ago as what we did diverge from horses, for example. And so, like I said, understanding key proteins such as cytochrome C allows us to be able to compare that particular protein in a variety of organisms that utilize that protein in the same way. And so basically, if we weren't able to compare those types of proteins, it would be because that organism didn't have it. And so if that organism didn't have it but another one did, it would probably infer that that organism couldn't do those same functions like aerobic respiration. And that's the reason we choose cytochrome C, is pretty much because all eukaryotic organisms conduct aerobic respiration. So it's a really, really good one. It's very common amongst all organisms, so it's easy to compare across species.